And I wanted to briefly show you what a three-factor ANOVA looks like. So I think what we'll do is we'll just skip over the assumptions because they're the same assumptions you've seen before for one-way ANOVA. And we can test them in the same way. So we can skip over that. And let's just in the last 6 minutes see what a three-factor ANOVA looks like. We've seen a one-factor ANOVA. We've now seen a two-factor ANOVA. Let's quickly look at a three-factor. So let's say we have three factors, A, B, and C. We have drug with 3 levels, low, and high dose. Sex with 2 levels, female and male. And let's say our third factor is age with categories, infant, young, and old. And let's say we're testing memory. That's our dependent variable. Let's not look at the numbers. Let's say that's what our data looks like. So now we have memory on the vertical axis, that's our dependent variable. We've got drug on the horizontal axis, that's one of our factors. In different lines, blue or red, we've got sex, that's the second factor. And the third factor, infant versus young versus old, we've plotted by using separate plots. So when you have three factors, there's no sort of 3D way of plotting it, so you have to plot it in a series of 2D slices. So that's what the data look like. And before we even look at the output, what does your brain tell you about what's happening? What my brain decides is that this drug really has no effect on memory in male mice, regardless of whether they're infant, young, or old. These orange lines are basically flat in all three of these plots. Placebo, low or high dose, it's the same memory score in infant mice, in young mice, and in old mice. Whereas this drug does have an effect in female mice. In infant and young and old mice, it increases the memory score. That's the main thing that pops out for me. The other thing that pops out is, in general, all of these data points in the old panel seem to be shifted up compared to the data points in the young, which are shifted up compared to infant. So the whole chunk of data seems to kind of increase in young mice and then increase again in old mice. In other words, there's a general trend for memory score to increase with age. Setting aside all this drug and sex stuff, right? When you have a three-way ANOVA, you now have 7 omnibus tests. In one-way ANOVA, you have 1 omnibus test, the effect of the factor, the main effect of the factor. In two-way ANOVA, you get 3 omnibus tests, main effect of A, main effect of B, and the A by B interaction. In a three-way ANOVA, you get 7 omnibus tests. It's kind of painful. You get the main effect of A, the main effect of B, the main effect of C. You get the A by B interaction, the A by C interaction, and the B by C interaction. So you get 3 two-way interactions, and you get 1 three-way interaction, A by B by C. So it's a little bit hard to think about what is a three-way interaction. The main effects are the same as before. The main effect of drug is the effect of drug when you average over sex and you average over age. The main effect of sex is when you average over drug and age, and so forth, right? The main effects are the same as before. These two-way interactions are kind of a combination of an interaction and a main effect. The interaction of drug by sex is testing how drug influences memory for different sexes when you average across age, ignoring the other factor. And the sex by age interaction is testing how memory differs across sex and age, averaging over the drug. So you average over the other factor that's not in the interaction. And then the three-way interaction, there's kind of multiple ways of thinking about what it means. The way I think about it is the three-way interaction tests the idea that one of the two-way interactions differs across levels of the third factor. So a two-way interaction is when the effect of one factor differs across levels of the other. A three-way interaction is when a two-way interaction is different depending on levels of the third factor. How would a two-way interaction be different across levels of the other factor? It could be that there is a two-way interaction for some levels and it goes away for the other levels. It could be that the two-way interaction reverses in some levels of the third factor. It could be that it just gets bigger or it gets smaller. It changes in some way. So it's a bit hard to pin down all of the examples because there's a huge number of ways that they could be different. Here's an example of a way that a three-way interaction could emerge. So I kind of changed the data. So now for infant mice, the drug has no effect for males or for females. But for young mice and old mice, the drug has an effect for females, but not for males. So this is the two-way interaction, right? No effect for males, yes effect for females. The two-way interaction is the same for young and old, but that two-way interaction disappears for the infant mice. So that's a three-way interaction, because the two-way interaction depends on the level of the third factor. The two-way interaction between sex and drug is different depending on the level of age, old versus young versus infant. The two-way interaction between drug and sex is present for old mice and young mice, but absent for infant mice. Does that make sense? It's a bit hard. I mean, that's one example, but the ways in which a two-way interaction can be different is almost infinite, right? You can think of drawing a whole laundry list of ways in which a three-way interaction could appear. The analysis plan for a three-way interaction, a three-way ANOVA, is the same concept for a two-way ANOVA. You start with the three-way interaction. That's the first thing you look at. And if the three-way interaction is significant, it's not here. If the three-way interaction is significant, you don't look at anything else. You don't look at any of the main effects. You don't look at any of the two-way interactions. And the reason is, all of those things, everything is up here. All three main effects, all three two-way interactions, they all involve averaging over something. Right? Each of these main effects involves averaging over two other factors. Each of these two-way interactions involve averaging over a third factor. If the three-way interaction is significant, it's telling you that everything matters. And so why would you average over anything if the three-way interaction is telling you that each factor has an independent influence? So if the three-way interaction is significant, that's what you look at. You try to do analyses, follow up. What is the nature of that three-way interaction? If the three-way interaction is not significant, then you jump to the two-way interactions. And then you treat each of them kind of like a two-way ANOVA. And if one of those two-way interactions is significant, then you dive into it and you figure out what's going on, either using simple manifest tests or pairwise tests, whatever. If one of those two-way interactions is significant, like let's say here the drug by sex interaction is significant, does it make sense to look at the drug or sex main effect? No, it doesn't. The drug by sex interaction is telling you that the effect of the drug on memory is different depending on the level of sex, male or female. So it wouldn't make sense to look at the drug main effect because it averages over sex and age. It wouldn't make sense to look at the sex main effect because it averages over drug and age. What about the age main effect? That's okay to look at. Age doesn't appear in the drug by sex interaction. The drug by age interaction is not significant. The sex by age interaction is not significant. The drug by sex by age interaction is not significant. Age doesn't appear in any interaction that's significant. And so it's okay to look at the age main effect, because its effect on memory is not modulated by any other factor. If it were, then it would appear in one of these interactions that's significant. The only significant interaction here is drug by sex. And so in this case, I would look here, the three-way interaction is not significant. Good. I would look at each two-way interaction. Those two are not significant. That one is drug by sex. So I would dive into that interaction and figure out how does the effect of the drug differ by sex. And just looking at the graph, you can tell what the result would be. And then I would look at the main effect of age, because the main effect of drug and sex are canceled out because the drug by sex interaction is significant. And what's the main effect of age? It's the effect of age on the drug. The effect of age on memory. When you average over levels of the drug and over sex, it's basically looking at all these data points versus all those versus all those. It's what I said at the beginning. It's this effect of memory increasing across age, irrespective of the drug condition or the memory condition, or the sex condition, sorry. So, it's true. A three-way ANOVA is a bit of a monster. It's a lot more complicated to analyze. It also involves a lot more data. Where are we? We don't need to look at that. Where do I have it? Well, maybe I didn't point it out. But let's just consider, if we have our three-way ANOVA is, where is it? Yeah, here we go. It's 3 drug levels by 2 sex levels by 3 age levels. So it's 3 times 2 times 3 is 18 different groups. And if it's between subjects, that means 18 groups of subjects. They're all independent from each other. Let's say we have 5 subjects in each group, which isn't that many. Then our total number of participants is 18 times 5, which is 90. So a huge number of participants for 3 factors that are fully crossed from each other. If we wanted 50 participants in each group, then we would need 900 participants in our experiment. It's huge. It just blows up in terms of the size when you add that 3rd factor. It's possible in principle to have a 4-factor ANOVA or a 5-factor ANOVA or a 15-factor ANOVA. It would be psychotic to do that. Both because it would be a giant experiment and it would be like a Kraken monster to analyze. But it's possible in principle. I don't know if I've ever seen an ANOVA that has more than 3 factors out there in the wild. It might exist, I don't think I've seen it. It's just not a good idea. We were talking about, I forgot, we were talking about earlier in the break, sort of the idea that, well, when you do science and you want to know what affects your variable, you could come up with a whole laundry list of things that you might want to vary in an experiment. So how do you can't do an experiment with 10 factors in it. What do you do? And it's true, you can't ever have the perfect experiment that controls for everything. So you kind of have to decide, what you're going to focus on, control what you can, vary what you can, and then in your discussion section, you have a section called limitations, where you talk about the other things that might affect the results, the other factors that might vary, and you spitball about future experiments and how they might address that. Okay, so I think we should stop because we're at 2 o'clock. I did skip over that kind of quickly. We could look at it again at the beginning of next week if we need to. I'm not going to ask you to analyze any 3-factor ANOVAs on the exam. That's not going to happen. So do have a look at this so you know about it. There might be 1 or 2 questions about 3-factor ANOVA. Like, for example, this kind of thing, if you have a 3-factor NOVA with this many levels and that many levels and that many levels, how many groups do you have? Infinite. So if you remember that, then you'll do well in that question. That question that might be on. Okay, that's all. See you next week.